Hi friends, welcome to biologyexamsfory.com. Today we are going to discuss about a very interesting topic, that is the difference between cyclic photophosphorylation and non-cyclic photophosphorylation in detail. And this is the most important reaction that is responsible for life on this planet, and it forms a part of light-dependent reaction of photosynthesis, and this reaction evolve oxygen and also convert the sunlight energy into chemical energy in the form of ATP and NADPH, and that is utilized in light independent reaction for the synthesis of glucose, and that will directly or indirectly feed all organisms in the planet except some bacteria. So first of all, starting with why this name photophosphorylation. As you all know, the site of photosynthesis is the organelle called as a chloroplast. Chloroplasts contain many membranous sacs that is stacked one above the other, which is called as grana. This individual sac is called thylakoid, and the first point, the site of light dependent reaction, is the thylakoid membrane. All the photosystem and electron carriers are located on the thylakoid membrane. This is the thylakoid membrane where photosystems are organized, and you can see the electron carriers, and this is the stromal region, and this is the thylakoid lumen where photolysis occurs, and this is the stromal region where ATP synthesis occurs. The reason for calling this reaction as photophosphorylation, here it is a sunlight driven process that provide energy for phosphorylation or conversion of ADP to ATP, and that is a sunlight driven process. That's why this is called as photophosphorylation. Let us zoom in this region for a detailed discussion. So this is the thylakoid membrane with photosystems and electron carriers. So we are discussing the non-cyclic photophosphorylation, and that's the most common pathway in higher plants that consists of both photosystem II and photosystem I. First of all, starting with photosystems. Photosystems are light capturing complexes consisting of antennae molecules that receives light, that is made up of accessory pigments like chlorophyll B, C, D, carotenoids, etc. And there is a reaction center, which is a chlorophyll A molecule. What is happening is this light energy is received or captured by the accessory pigments, and this energy is transferred by resonance transfer. It's a kind of vibratory transfer, and ultimately this energy is transferred to the reaction center chlorophyll molecule, where the electrons are excited from ground state to the excited state. There starts the electron flow or electron transfer. Then electrons are moved through different electron acceptors like plastoquinone. So the first point, there are two photosystems involved in light dependent reaction. So the electrons ejected from this photosystem that travels through electron carriers like plastoquinone. During the movement of electron, the movement of electron is from high energy level to low energy level. Therefore, some amount of energy is released during the electron flow or electron transfer, and that energy is utilized to pump H+ or protons from the stromal region to the thylakoid lumen region, and this creates a gradient or more number of H+ in the thylakoid lumen. That's called as a gradient. First point, there are photosystems involved that captures the light energy and causing the excitation of electron of chlorophyll A or reaction center molecule. Second point, during the electron transfer, the energy released is utilized to pump H+ from stromal region to the thylakoid lumen region, and this is happening through cytochromes. Cytochromes are proton channels. So this creates a gradient, and this electrons moves to plastocyanin and finally reaching PS1. Here also the light energy, these electrons are in little energy state, and this is further excited by sunlight and moves to ferredoxin, and there is an NADP reductase enzyme that will convert NADP to NADPH. It's a reductase enzyme, therefore it will add H+, and finally making NADPH, which is the output of non-cyclic photophosphorylation, NADPH and ATP. In PS2, there is an electron hole. Electrons are excited. This is to be refilled, and there is a process which is called as photolysis of water or water splitting that will release H+ and oxygen, and this oxygen is evolved from plant, and that is utilized by all other aerobic organisms. And Here also the H plus is released. This also creates a gradient or more number of H plus in the thylakoid lumen. So the main point to remember is the gradient is created by the energy released during the electron flow that is used to pump H plus from stroma to thylakoid lumen. And the second way is by the hydrolysis of water, this H plus is directly supplied into the thylakoid lumen. Therefore, H plus in thylakoid lumen region increases considerably. There is a tendency to make an equilibrium, but there is no way out for this H plus through this membrane. The only way out is a protein complex which is called as ATP synthase. When this H plus moves through ATP synthase is an enzyme which is having active site for ADP and phosphate. During the proton flow, ADP and phosphate combine to form ATP. The output of non-cyclic photophosphorylation is a synthesis of NADPH and ATP. In summary, in light-dependent reaction, the energy of sunlight is trapped by the photosystems and that is converted to chemical energy in the form of ATP and NADPH, and that is utilized to convert carbon dioxide to carbohydrate in light independent reaction or dark reaction. Hope things are clear. Now moving into the difference between cyclic and non-cyclic photophosphorylation. First of all, starting with why this name. So this is another diagram of cyclic photophosphorylation. Only one photosystem is involved, that is P700 photosystem 1. Sunlight energy that is trapped by accessory pigments and that is transferred to reaction center where electrons are ejected, and there is a primary acceptor and passes through ferredoxin plastoquinone. There is a cytochrome complex where H plus is transferred to a proton channel, and ultimately this electron flow is cyclic and it reaches a PS1. That's why it is called as cyclic, and electrons return to PS1. Whereas in non-cyclic photophosphorylation, two photosystems are involved. The electrons that is ejected from photosystem, it moves through primary acceptor which is often called as phaophytin, then plastoquinone, cytochrome complex, and ultimately reaching PS1. It is further energized by the sunlight and ultimately reducing NADP to NADPH. Here the flow is non-cyclic or linear. Electrons moves from PS2 to PS1 and ultimately reducing NADP to NADPH. That's why this is called as non-cyclic. Second difference regarding the output and photolysis. In non-cyclic photophosphorylation, ATP and NADPH are produced. So two energy-rich molecules are produced, ATP and NADPH. And that's why this is predominant in higher plants. And that's a process called as photolysis of water. Here the electron flow is not cyclic, therefore the electron that is ejected from photosystem 2 should be refilled, and that process that is refilled by photolysis of water. It provides oxygen and also H plus releasing electron, and that is taken up by the photosystem 2, filling that electron hole or electron gap in photosystem 2. Whereas in cyclic photophosphorylation, only ATP is synthesized. 2 ATP is produced. It is predominant in bacteria. There is no photolysis of water as electrons are returning back to PS1. There is no evolution of oxygen, therefore involved in anaerobic anoxygenic photosynthesis. When there is shortage of NADP+, this cyclic photophosphorylation is preferred even in higher plants. Thank you so much for your support. You are with biologyexams4u.com.